Record. Okay, I almost forgot that. Because I'm going to put this up in YouTube in case somebody missed it or you want to look at it again. Okay, so I handed out these model kits, and it's called ball and stick models, and there's round pieces of plastic in there of different colors that represent different atoms. And we work with only 4 atoms, the big 4: carbon, oxygen, nitrogen, and hydrogen. That's it. Like about 95% of your body or the bodies of living things are composed of molecules that are made up of those 4 atoms. So, and they're different colors. And then there's bonds, there's little plastic sticks like that are bonds that we put the atoms together to make these molecules. Okay. So, and then we go along and we, can I move this? Okay, and then we go along and we fill out spaces here. We answer questions, we do some drawings down here, and so on and so forth. And we just fill this out as we go along, and then answer some questions at the end, and then we're all done. And then somehow he turned into me to get graded, right? And this is an individual lab. Everybody does their own sheet. And interrupt me if I'm not clear on something, if you're not sure. Don't worry about interrupting me.
Do you want us to turn it in whenever we come back to college, or do you want us to give it to you through like a Word document?
Either one. It's not really due like right now. If you want to hang on, if you want to make an electronic copy and send it to me, that's fine. If you want to hang on to it until we come back onto campus the week after next, that's okay. You know, whatever you want to do. All right. So here's the objectives: learn the definition of molecule, practice drawing molecules, make models of selected molecules. We'll talk about that, and we'll say what we would have done had we been on campus. To learn what single, double, and triple covalent bonds are, and actually look at how there are 2 types of covalent bonds, what a covalent bond is, and the 2 types. To be able to convert a molecular formula to a structural one. Remember, the molecular formula is what atoms are participating in a molecule and how many of them there are. That's called a molecular formula. But then you gotta be able to convert that to what's called a structural formula. That is, how are the atoms attached to each other? And that is really important, how they're attached to each other, because you can have 2 molecules that have the same molecular formula, but different structural formulas. These are called isomers. And the way the atoms are put together is really important to the way these molecules work in biological systems. Okay. And then learn what a macromolecule building block is, and to make molecular models of building blocks. So most of this lab is chapter 2, essential chemistry, but then we get into a little bit at the end of it, chapter 3, molecules of life, macromolecules, right? All right. Starting with what is a molecule? All right. At least 2 atoms connected by a chemical bond. That's what a molecule is. They can be really simple, like the oxygen you're breathing in right now. You're breathing in the oxygen molecule, yeah, O2. It's 2 oxygen atoms connected by a chemical bond, actually connected by a double covalent bond. Okay. Or they can be really complicated, like the macromolecules. All right. So here's a molecular model kits, plastic balls with holes in them. The balls are different colors and they represent different atoms. The black balls were carbon, the red balls oxygen, the blue nitrogen, and the white hydrogen. There were a couple other colors in there, but we didn't use them. Somebody's in the waiting room. Okay. So the balls have holes in them. And they have different numbers of holes in them. So why is that? What are we going to be putting into the holes? What holds together atoms in a molecule? Anybody?
Is it like the chemical bonds?
Bonds, yeah, chemical bonds. I was just mentioning the molecule. We're going to be, these little plastic sticks are going to be put into the holes and they're going to represent chemical bonds, covalent bonds. All right. Okay, and the bonds, the white and gray pieces of plastic represent the covalent bonds. Small white ones are only to put attaching hydrogen to other atoms, and then you have inflexible pieces of gray that bond, and then longer flexible ones. They represent single covalent bonds. And so what is a covalent bond? Well, it's a pair of shared valence electrons. So remember, bonds, molecules either give up or take on electrons to form a bond. Those will be ionic bonds. Or they share electrons. Why do they make these bonds? And how many of the bonds does each atom make? Well, what I do was I would ask people to pick up the black balls, they represent carbon, and say, how many holes does it have? 4. And then they would pick up the red balls and say, how many holes does it have? 2. Pick up the white balls, which are smaller than the rest. How many holes does it have? 1. And the blue ball has 4 holes, but we're going to make believe it only has 3. And this is the number of bonds these atoms make. Question is, why? Why did they make that number of bonds? And when you were going to put these molecules together, carbon has to make 4 bonds. Oxygen 2 bonds, nitrogen 3 bonds, hydrogen 1 bond. And so you have to make molecules and all the holes have to be filled with bonds, right? And let's talk about why that is. I'm going to stop sharing this, and I'm going to share some stuff on the web with you. For one thing, a periodic table of the elements. All right, let's pull up a periodic table. All right, let's share this one. Wait, is that correct? Yeah. Okay. Here's periodic table of the elements. Okay. So let's look at carbon. Topic number 6. That means it has 6 protons and 6 electrons. But here is the thing: how are the electrons arranged around the nucleus of atoms? They are put into what are called shells. The first shell, second shell, third shell, however many shells you need to fit all of the electrons in, right? And here's the thing. shells can only hold so many electrons. As a matter of fact, the first shell holds 2 electrons, only holds 2. Why? I have no idea. It's just, this is just something you got to accept. This is how the world works. You know, the first shell holds 2 electrons and no more. Second shell and every other shell holds 8 electrons, a maximum of 8. Right? And so we're going to follow what's called the octet rule, which is true for every shell except the first. Right? And here's the thing: the electrons in the outermost shell. Well, the outermost shell is called the valence shell, and those are the electrons that participate in chemical bonding. Those are the ones that are given away or taken up or shared, or the valence electrons. Okay. Now, so it's important to know how many valence electrons these atoms have. So you look at carbon, that's 6 total, right? So how many valence electrons does it have? Well, there's 2 in the first, and that leaves 4 left over to make 6. It's got 6 electrons, 2 in the first, 4 in the second. Okay, so it's got 4 valence electrons. And what these atoms want, somebody's still in the waiting room. Wait a minute. Are they? Hold on, I gotta stop sharing a moment and look at the waiting room. There's nobody in the waiting room. Wait a minute, hang on just a second. Somebody's stranded, I think. Adrian Grove, okay. Let me send her an invite. Right there. Oh, there she is. All right, let me go back to the periodic table. Okay, so what atoms want, and why do they want this? I don't know. What atoms strive to do is get filled valence shell. They want a filled valence shell. Happiness for an atom is a filled valence shell. And they'll do anything to get it. Like I said, why? I don't know. But it's really good that they want this, because if atoms didn't want to fill valence shell, there wouldn't be any chemistry. There wouldn't be any bonding between atoms, and we wouldn't be here talking to each other. The Earth would just be a big ball of elements, single elements with no chemistry. So it's really great that atoms want to fill valence shells. Okay. So carbon has 4. How many does it want? 8. Because its valence shell is the second shell, and that holds 8. So it wants to get 4 more electrons. So to make deals with other atoms to share electrons and get 4 more, right? You know what I mean? So you can always tell how many bonds you expect an atom to make by taking its valence electrons and subtracting it from 8. And that's how many it's going to make. For example, oxygen, atomic number 8. 8 total electrons. How many valence electrons? 2 in the first, 6 in the second. How many does it want? 6 from 8 is 2. It makes 2 bonds. That's it. See? So that's called the octet rule. And it's true for every atom except for hydrogen, because hydrogen's valence shell is the first shell, and it only holds 2. So it just wants 1 more electron, right? Okay, these are, here's the thing. You could always mathematically figure out how many valence electrons an atom has if you just point to it and look at what number it is. wish I had a larger one here. Oh, well, silicon, topic number 14. How many valence electrons does it have? You can figure it out mathematically. 2 in the first, that leaves 12. 8 in the second, that leaves 10. 4 more left over. It's got 4 valence electrons, like carbon. As a matter of fact, that's why it's in this column here. So here's the thing, how you can read the periodic table. And I think I'll be going to be, on the exam, I'm going to have to find some way to let you look at a periodic table in case I ask questions like this on the exam. So here's the thing, we're going to look at 8 columns. This column, 2, skip over these middle guys. They don't play the same way. So we're like, some of them are important biological atoms like iron and so on, but we're not going to, so we're going to look at this column, 3, 4, 5, 6, 7, 8. They are in those columns because all of the atoms in those columns have the same number of valence electrons. So all these guys have 4, 5, 6, 7 valence electrons. 1 valence electron, 2 valence electrons. That's why they're in those columns. So you can just look at the periodic table elements and tell how many valence electrons these atoms have, or you can figure it out mathematically by looking at how many electrons they have. Okay. Now we're not going to do, we're going to look at covalent bonds here. And generally these guys, so column 3, 4, 5, 6, all these guys generally make covalent bonds. Because it's, remember, ionic bonds are you give up electrons or take on electrons. Well, it's tough to take on 4 or 5 or however you need. It's easier to share. So those atoms generally make covalent bonds. They sometimes make ionic bonds, but generally it's covalent. But these 2 columns and this column, 1, 2, and 7, generally make ionic bonds. And here's why. Here's column number 1. How many valence electrons? 1. Ooh, they're really unhappy. All right. And here's the thing about having one valence electron. It's also part of nature is that if a shell doesn't have any electrons in it, it disappears. So let's look at sodium, atomic number 11. 1 valence electron, 2 in the first, 8 in the second, 1 in the third, which is its valence shell. Really unhappy. Oh, if I could just get rid of that 1 electron, the third shell will disappear. My valence shell will be the second shell, and that's got 8. So I'd be happy. So sodium is really anxious to get rid of that 1 electron. Matter of fact, so anxious, it's dangerously active. Sodium at standard temperature and pressure is a metal. It's an explosive metal. It's very dangerous. It comes into contact with water, it explodes. It happened one time in taking a organic chemistry class. We made elemental sodium as a byproduct, and somebody dumped it down the sink and it exploded all over the room. So it's dangerous. All right, it will violently get rid of that electron if it needs to. Okay. Then over here, atomic, these are column 7. How many valence electrons do they have? 7. Ooh, so close. There's 1 more. But in this case, I want to get an electron. So, and chlorine also is a dangerous element. I mean, it's standard temperature pressure. It's a green gas, a deadly poisonous green gas. All right. So we got an explosive metal, poisonous gas. All right. You put those together, and they're made for each other. Because sodium wants to get rid of 1 electron, and chloride wants to gain 1. So if chloride gains 1, it gets 8. Sodium loses one, it gets 8 in its valence shell, and everybody's happy. And they make an ionic bond between them, and it's a molecule called sodium chloride, NaCl. What's NaCl? You use it every day, probably.
Salt.
Salt. You shake it on your food. It's salt. How innocuous is that? You know, except you've got high blood pressure. You know, as a matter of, and so here's what happens when atoms give up or take on electrons. They get profoundly changed. They are no longer, when sodium gives up that electron, it's no longer sodium. It's sodium ion, and it has a positive charge. Chlorine, no longer chlorine when it gets that extra electron. It's now chloride ion, and it's got a negative charge. Positive ion, so it gets a full charge. Positive ions are called cations, negative ions are called anions. Why does sodium get a positive charge or gives up an electron? Well, remember, these atoms are electrically neutral because they have the same number of protons in the nucleus as electrons in their shells. And protons are positive, electrons are negative. They cancel each other out. So there's no charge on these atoms. But once sodium gives up an electron, now it still has 11 protons in its nucleus, 11 positive charges, but it only has 10 negative charges now. So it's got an extra positive charge. So it gets a positive, Na+. Chlorine, it's 17 positive charges in the nucleus, 17 negative charges in its shells. Gives up one, now it only has 16. Oh no, gains one has, so gains it has 18. So it has an extra negative charge. 17 positive, 18 negative. So it gets a negative charge. See what I mean? So so you need to know what an ion is. When an atom gives up electron or gains an electron, it becomes an ion. Okay, and that's true for all of these guys and all of these guys. They all want to give up one. They all want to gain one. So they make molecules with ionic bonds like lithium fluoride, potassium bromide, potassium chloride. All of them make ionic bonds. So column 1, column 7 make ionic bonds with each other. Right? Also, column 2, they have 2 valence electrons. They would like to give up the 2 also. So like magnesium, would like to give up 2 electrons. So it makes, so let's use chlorine again. So we can make a molecule called magnesium chloride. But magnesium wants to give up 2. Each chloride wants to gain 1. So magnesium makes a deal with 2 chlorides. So magnesium chloride is MgCl2. And magnesium becomes a double cation, 2 positive charges, because it's given up 2 electrons. So it has 2 more positive charges than negative charges. Okay, so we're not really doing ionic bonds in this molecular model kit, but I want you to know about ionic bonds. And I talk about it in the video lecture about ionic bonds. And they're pretty weak. They're weaker than covalent. You put those molecules in the water and they break apart. They dissolve in water. You put salt in water, what happens? It dissolves because the ions break apart. Okay, we got that, all right? So let's go back to the covalent bonds and the lab paper. All right, where is it? Okay. Wait a minute. And remember, covalent bonds, there can be single, double, or triple covalent bonds. And we'll show you that in the next part with small molecules. Here we have small molecules. And what you do is, and you write out the molecular structure for this small molecule, and then you draw the molecule, how it looks. And this is the, okay, so let's start with water. What's the molecular formula for water?
H2O.
H2O. One oxygen, two hydrogens. How are they put together? And let's look at a ball, how we represent the ball and stick models. Hang on a second, let's go back to the web here. Okay, so remember the structural formula is how the atoms are attached to each other. So you have two hydrogens. So how about if you attach one hydrogen to the next, to the other hydrogen, and you attach the oxygen to a hydrogen? Would that be correct? If the two hydrogens were attached to each other, and the oxygen was attached to one of the hydrogens, would that be a correct structural formula for water? Well, can hydrogen be attached to another hydrogen and to an oxygen?
No.
No. How many bonds does hydrogen make? One. And you wouldn't, and oxygen always makes two. So the structure of water is like this. Oxygen bond, and this is how you draw them. You draw, you don't have to draw the balls. You could just write O, and then you'll draw a line between the O and the H, and that represents a single covalent bond, and to a hydrogen. So here everybody's happy. Hydrogen is sharing one pair of electrons with oxygen. Each hydrogen is sharing one pair of electrons with the oxygen, and oxygen is sharing two pairs of electrons, one each with hydrogen. So everybody's got filled valence shells and everybody's happy. Okay. Then if you were, okay, so if I was going to go to, so we know what water is. Let's go next to methane. The molecular formula for methane is CH. Well, it's one carbon. So how many hydrogens does it have? 4, CH4. So there's, okay, methane images. There's methane, CH4, right? And then ammonia. And methane is a flammable gas. It's part of natural gas. Household chemical ammonia. Wait a minute. I don't want the Lewis structure. Ammonia. NH3. And remember, nitrogen is atomic number 7, 2 in the first, 5 in the second, which is the valence shell. 5 from 8 is 3, makes 3 bonds. Now, but here's the thing. If you did see this blue ball that represents nitrogen in the kit, it would have 4 holes. Because As they always say, rules are made to be broken. And the octet rule is broken sometimes. Sometimes atoms rarely break it. Some atoms rarely break it. Some atoms break it pretty often. Some break it all the time. Phosphorus, for example, breaks it all the time. Nitrogen breaks it every once in a while. It can pick up another hydrogen and actually become what's called the ammonium ion. Well, this is like no, that's a Lewis diagram. So you can get NH4 and yeah, NH4, the ammonium ion, it has a positive charge because it's got an extra hydrogen, and that's what is a hydrogen? It's a proton, so it gets a positive charge. But we were not going to let it do that in the lab. We were always going to stick to the octet rule and only let it make 3. Okay. Okay. A little bit more difficult now would be carbon dioxide. So what's the molecular formula for carbon dioxide? Tells you what it is. Carbon 1, dioxide 2, so CO2. So I would ask everyone, take 1 carbon, 2 oxygens, make a legal molecule, fill in all the holes. How are you going to do that? So carbon has to make 4 bonds and each oxygen has to make 2. How are you going to do that? And I usually say you have to be flexible in your thinking because you got to move from the rigid gray sticks to the flexible gray sticks.
Does each carbon double bond to the oxygen?
Well, each oxygen double bonds to the carbon. Yeah, let me, right here. There it is. That's CO2. So atoms can share more than 1 pair of electrons. This is called a double covalent bond. Okay? And everybody's happy because carbon has 1, 2, 3, 4 bonds. Each oxygen has 2. Everybody's happy. Okay, well, we know what carbon dioxide is at standard temperature and pressure. It's a gas, right? And what's the source of carbon dioxide in your room right now?
Us?
Air.
Yeah, you are. It's that's something we breathe out. Every exhale breath has got carbon dioxide. It's a waste product of metabolism. So we get rid of it in our exhale breath. It's also a greenhouse gas. We're putting more of it. It's supposed to be like only 0.04% of the atmosphere, but we're putting more in it and it captures heat. And so it is a greenhouse gas and we want to try to limit that much of, when they say we're putting carbon in the atmosphere, that's what they're talking about. And also that comes out of your tailpipe. When you drive, what comes out of your tailpipe is carbon dioxide. It's a byproduct. Whenever you burn anything, you make carbon dioxide. And your food, you actually burn it. It's just like you burn gasoline in a car. It's essentially the same chemical process, but it's done explosively in a car and it's done in small, careful steps inside living things. Okay. Now, one other thing that I added was hydrogen cyanide. Okay, hydrogen cyanide is it's a very simple molecular formula, HCN. 1 hydrogen, 1 carbon, 1 nitrogen. And I would ask the class, put these together, make a legal molecule, leaving 1 hole on the nitrogen open. And how are you going to do that? This is the only way it works. Triple covalent bond. So carbon and nitrogen make a triple bond. They're sharing 3 pairs of electrons. And again, everybody's happy. And then it shares 1 pair of electrons. Carbon shares 1 pair of electrons with the hydrogen, 3 pairs with the nitrogen. It gets 4. Nitrogen gets 3. Hydrogen 1. Everybody's happy. See? So you can have single, double, triple covalent bonds. How about quadruple covalent bonds? No, they don't exist. And of course, not all atoms can make triple bonds. Oxygen can't make a triple bond because it only makes 2 bonds. Okay. And what is hydrogen cyanide? At standard temperature and pressure, it's a gas. And like chlorine, poisonous gas, even more dangerous than chlorine. I mean, it's a really, really deadly gas. And how did we used to use it? Well, one state used it a long time ago, California. When California had a death penalty, they had a gas chamber, and they would use this gas to legally kill people. But that's not done anymore. Now, any state that has a death penalty, uses lethal injection. They don't use gas anymore. Or even worse, the electric chair. That was really bad. Anyway, so let's stop with that and then go back to the molecules paper. So you draw out those molecular and structural formulas for those small molecules. Then it asked, can more than one structure be drawn for each molecular formula? Can you draw water any other way? Or methane or carbon dioxide? Or there's just this one structural formula for those? Anybody?
Just one.
Just one. There's only one way you can draw water, carbon. Now when we get the larger molecules, you find out that there are different ways, and this is where the isomers come in. Like, for example, the larger molecules. Here's a larger formula, slightly larger, H6C2O. And then I'll ask the class, make a legal molecule using your model kit with 2 carbons, 1 oxygen, and 6 hydrogens. So all the holes are filled. And the hint would be take the carbon and oxygens, put them together any way you want, and then see if you have enough holes to put 6 hydrogens in. And there are actually two solutions to this. One of the solutions is a very popular molecule. Let me, again, stop sharing this and go back to the web here. Okay, let me share this screen. So here's one answer to that, and as I said, it's a very popular molecule, perhaps too popular. I partake a little bit too much of this particular molecule, and some of you in the room are not old enough to legally partake of this molecule. So what is this? It's alcohol. It's alcohol. That's one of the solutions to that. So two carbons, 1 oxygen, and 6 hydrogens. So you put the 2 carbons together with the oxygen, and you get 6 O's. Now sometimes this OH here, this is a shorthand. There's a bond between those. Well, it's right here in the stick model. But when you draw it out, they just leave it out. and it's just taken for granted that the, you know, bond between the O and the H there. That's called a hydroxyl group, which we're going to get to later on. And then the other, but there's a, and I used to call this a little dog, because this little dog, we put it on a table, it's like a little dog. There's another solution though. No way. Those are two solutions. Ethanol on the left, something called dimethyl ether on the right. And in that way, so there's two ways you put the carbons and the oxygen together. Two carbons attached to each other and then the oxygen, or the oxygen attached to two carbons. And either way, you have 6 holes to fill in with hydrogens. So these are isomers. Same molecular formula, different structural formula, right? Okay. Then the next molecular formula under the larger molecules is H4C2O. So you have to take away 2 hydrogens. And now I say again, make a legal molecule. Take 2 hydrogens away and make a legal molecule. So how are you going to do that? And I usually say you have to be flexible in your thinking. I don't know what this molecule is named. There's several correct ways of drawing a structural formula for C4H2O, but what is required is a double bond somewhere. You have to have a double bond. Let's do I have something? Well, that's similar. I mean, there's no O here, but that would be a solution. So let's make believe there's an oxygen here, and the hydrogen is attached to that oxygen. That would be a solution. You need a double bond somewhere, and you only have 4 hydrogens. Now, double bond could be I'm talking too fast. Double bond could be between 2 carbons, or between the carbon and the oxygen. So there's a couple of different solutions, but you need a double bond. So here's the take-home message. If you haven't got enough hydrogens to fill up all the holes, you think double bond. Somewhere there has to be double bond. Okay? All right. Then the third one is C4H2O2. And now you have to add something. You got to add an oxygen. And it's really easy to do, but you still need a double bond because you still only have 400. So you keep the double bond, but you detach one of the hydrogens from a carbon, attach the carbon to the oxygen that you're adding, and then add the H to the oxygen that you've added, and then you've got, you know, a correct structural formula for that molecular formula. Everybody see that? So if we haven't got enough hydrogens to fill up all holes, think double bond. All right, let me stop sharing that and get back to the whoops. I hit end instead. Whoops. Oh, share. Okay. So then it's easy answer to the question down here. Can more than one structure be drawn for each of these molecular problems? Well, yeah. You can make isomers. And the more complicated the molecular formula, the more isomers you can make from it. Okay. So then we move down. and we start talking about organic molecule and an organic backbone. So what is an organic molecule? Well, sometimes life on Earth is called something-based life. One of the atoms is really important, and it makes up the backbone of all of these molecules. So what atom am I talking about? What blank-based life?
Carbon.
Carbon-based life. All life on Earth is carbon-based. Now maybe on other planets, they use a different atom, but on this planet, we use carbon. So the one thing about organic molecules is they're carbon-based. So you gotta have a carbon, at least one. But also they need one other thing. They need at least one carbon-hydrogen bond, or else it's not an organic molecule. So methane, organic? Yes, has a carbon. Alcohol, organic? Yes. But carbon dioxide, is that organic? Carbon-based, but no.
Organic if it comes from you, but it's also inorganic if it comes from a car.
Well, it's inorganic because
Okay.
it doesn't have a carbon-hydrogen bond. So even though it's carbon in this molecule, it's not organic. So carbon dioxide is not organic. It's inorganic. And you know, there's a whole branch of chemistry devoted to the chemistry of carbon, organic chemistry. And then there's inorganic chemistry. And actually, organic chemistry is more complicated, and that's the other of the 91 elements. But actually, organic chemistry is more complicated than inorganic chemistry because carbon is so versatile. It makes 4 bonds. It sometimes makes ionic bonds also, but it makes 4 bonds, and it binds to just about any of the other 91 elements. You can, and all of our big important molecules have the carbon, what's called a carbon skeleton. And you put, so that's why down here, I would ask everyone to take 6 carbon atoms, and don't worry about open holes, just put together 6 carbon atoms any way you want. You can make a straight line, you can make a branched chain, or you can make a circle. So actually, let me show you that. Go back to the end. Where are we? So types of carbon skeletons, like here, straight chain, branch chain, ring, anything. It's so that's one of the why carbon is so versatile, because you can make so many different shapes with it, like down here. And you got double bonds, single bonds, double bonds, sometimes triple bonds with carbon. So it's really versatile. Okay, so there's a reason why the carbon-based life. Okay, next thing, we go down to page 3 of the lab paper. We have functional groups, and we have 5 of them. And here's the thing: so these big important biological molecules have a carbon skeleton, but they have different roles. They do different things. And why do they do different things? Because of what? Because of arrangements of other atoms, particularly oxygen, hydrogen, and nitrogen, the other of the big 4. They are put together in certain ways called functional groups. And you add those functional groups to the carbon skeleton, and it changes things. It makes it do different things. So let me show you that. And the first functional group we talk about is the hydroxyl group, or the OH group. Okay. Okay, that is the hydroxyl group, this OH. And sometimes it's just written like this as OH, you know, the bond left out. Oops, that's not good. Well, that's the different functional group. Let me go someplace else. No, that's not. Getting ahead of ourselves. Here we go. OH. So here's the thing about adding an OH to a molecule, how it profoundly changes it. And now we're going to get into what's called polarity. So here's the thing. There's two types of covalent bonds. They're called full covalent bonds or non-polar covalent bonds. And then there are polar covalent bonds. Now, a bond between two carbons is a non-polar bond. And what that means is equal sharing of electrons. The electrons are equally shared, so there's no charge on either side. Also, carbon-hydrogen bond is also non-polar. Right? So if you have, let's say, a molecule called C2H6. So you have, I think that's butane. Let's see here. Okay. Let's make believe this oxygen, this OH group wasn't on this molecule. This is ethyl alcohol again. Let's make believe that's just a hydrogen. That I think is butane, and it's what's called nonpolar. It's all got nonpolar bonds. But this bond here, if you had an OH to it, profoundly changes it, because now you've made a polar molecule. Why? Because this bond here, actually a bond between the O and the H and the O and the C, and between O and anything, is called a polar covalent bond. Because oxygen has what's called a high electronegativity. It loves electrons. I mean, really. And it tries to take electrons away from other atoms. Actually, that's why oxygen is actually kind of a dangerous gas. You know, when it was first put on Earth, when photosynthesis was invented and oxygen was a waste product of photosynthesis, that was the first case of air pollution on Earth, because it's quite a dangerous gas, because oxygen strips electrons from other atoms. It's called oxidation, and it's dangerous. And we actually have, even though we need this stuff, and we later on when we get into cellular respiration, we'll learn that we use oxygen and its ability to take electrons to get rid of waste electrons in our own metabolism. That's what we use oxygen for. That's why we need to breathe this stuff. But it's also very dangerous. We have to we have to have special antioxidants to protect ourselves against the ravages of oxygen. That's one of the reasons why we age, damage from oxygen. Okay, so since the oxygen is trying to steal the electrons from the carbon, the electrons spend more time near the oxygen than they do near the carbon. This is unequal sharing. And since electrons are negative, there's what's called a partial negative charge next to the oxygen, and a partial positive charge over here. So you have poles, a polar molecule. Also known as a hydrophilic molecule, because they mix with water. Does alcohol mix with water? Mixed drinks. Oh yeah, of course it does. But butane would not mix with water. So in the segregation among molecules, nonpolar molecules hang out with each other polar molecules hang out with each other and they don't mix. Like oil and water, you put oil and water together. Oil is non-polar, it's called a hydrocarbon, as a matter of fact. Gasoline also is a hydrocarbon because it's all carbon and hydrogen. That's it. And that's a hydrocarbon and it's a non-polar molecule and it's hydrophobic. It's afraid of water, doesn't mix with water, right? So this concept of polarity is really important. Okay, so a carbonyl group is just a carbon double bonded with an oxygen, carbonyl. Just a double bonded, like I said, an oxygen double bonded to a carbon. It's a carbonyl group. All right. Oh, I'm sorry. Down below. I'm sorry. If you look down below, you got to answer the questions. In the functional groups, we got hydroxyl. So draw out a hydroxyl. You can just draw out, OH. And then you, farther down, it says which of these groups are polar or non-polar. It's a polar group. In fact, anything that has oxygen or nitrogen is a polar group because nitrogen acts just like oxygen in trying to steal electrons. Okay. And then what general types of larger molecules contain each of the functional groups? And that is, well, I showed you the ethyl alcohol. That's got a hydroxyl group, right? But there's other kinds of alcohols. There's alcohol with only one carbon, not two. That's called methanol. And that's some type of alcohol, but not the drinkable. Not unless you want to keep your eyesight, because it'll blind you if you drink the stuff. And then there's isopropyl alcohol, 3 carbons. And that's rubbing alcohol, also not a drinkable kind. Okay. Carbonyl is, put carbonyl down as a polar group also. All right. So the hydroxyl, what general types of larger molecules you put down alcohols, or you put down sugars. We are going to go on to sugars later on, and they got lots of hydroxyl groups. Okay. Then what we're going to do is take the carbonyl group and the hydroxyl group, put them together on the same carbon to make the carboxyl group. Whoops, oh no. Well, oh wait, let me do that. Sometimes carbons are replaced by angles. Later on, when we get into sugars and larger molecules, there's a shortcut way of drawing these like this. We just put the angle in here, and that's just taken for granted there's a carbon there, all right? Or you can keep the carbon. So that's a carboxyl group. Is it polar? Very polar. Well, larger types of molecules, you find lots of carboxyls. And here's acids. Now you've heard the term acid and base, and we have to define what an acid and base is. And an acid is any molecule that adds hydrogen ions to a solution. Hydrogen ions are H+. It's a proton, it's a naked proton, that's a hydrogen ion. And it's very reactive. And so this is called a pH, when we measure pH of a solution. And why this is acidic is that, see this bond right here? Well, sometimes the oxygen actually does steal electrons away from the hydrogen. And the hydrogen breaks off, and the oxygen has an extra electron gets a full negative charge now, rather than a partial negative, it's a full negative. And the hydrogen becomes an ion, the hydrogen ion. And so if you add this molecule to water, some of these bonds will break, and you'll increase the hydrogen ion concentration of a solution, and that's acidic. We have high levels of hydrogen ions, that's an acid. So you put down organic acids for the general types of larger molecules that contain a carboxyl. All right. Then you go on to amino. Now this one has nitrogen in it. So they are together. Carboxyl over here, amino over here. So amino group, NH2. And is it polar? Yes. Nitrogen makes things polar as well as oxygen. In what larger types of molecules do you find lots of amino groups in? The opposite of acid, bases, organic bases have got, because remember, nitrogen can sometimes make a fourth bond. In fact, here it is right here. Nitrogen sometimes picks up another hydrogen. And it has 4 bonds and has an extra proton with the hydrogen, so it gets a positive charge. So bases reduce the hydrogen ion concentration of the solution by soaking them up. So that's why this is a base, right? And then finally, methyl group. And that's CH3, that's it. And is this a polar or non-polar? Well, does it have oxygen and nitrogen in it? No. It's just carbon hydrogen. Those are non-polar covalent bonds, and it's the only one of the functional groups that's not polar, it's non-polar. And where do you find, the bottom, it says, where do you find a lot of larger molecules, a lot of methyl groups? Well, think of what's a big non-polar molecule you can think of at home. I mentioned it before. You put it into water, it doesn't mix. What would it be?
Oil.
Oil, yeah, any kind of oil. Or fat, take fat, oil, gasoline, pour into water, doesn't mix. It's, fat or oils, you find a lot of methyl groups. Okay, now you have phosphate there just above it. You can leave phosphate blank. We didn't do anything with phosphate. Okay? Okay, then let's stop sharing that. Go back to the okay, now I'm going to get into macromolecules a little bit and building blocks of macromolecules. So a building block is called a monomer, medium-sized molecules that make up very large molecules called macromolecules. General categories of these medium-sized molecules: sugars, glycerol, fatty acids, amino acids, and nucleotides. And they make up carbohydrates, lipids, proteins, and nucleic acids. So, and 3 of these, carbohydrates, proteins, and nucleic acids, are what are called polymers. They're long strings of these either identical molecules or similar molecules put in long strings, right? So they're organic polymers. Actually, plastic is an inorganic polymer, but these 3 out of these 4 macromolecules are organic polymers. So, so one of these would be called a monomer. 2 of them would be called a dimer, 3 of them attached are called a trimer, and 4 or more becomes a polymer. And they're put together in reactions called dehydration reactions. And they're all put together this way. And what dehydration means, what do you take away when you dehydrate something? Water. You take water away, and you can attach these monomers one to another in long strings. Okay, so the first monomer we're going to look at is sugar. Biologically important sugars have 5 and 6 carbon skeletons. That's why I would have you make a 6 carbon skeleton, because we're going to make a sugar. So you take off all functional groups from your 6 carbon skeleton, make an unbranched chain of carbon atoms, add a hydroxyl group to every carbon, and then link the 1st and 5th carbon in a circle by connecting them by the 5th carbon's oxygen. You will lose 2 hydrogens in this process. Then you draw the molecule. And there's a simple way of drawing, actually it's down here, a little simpler way of drawing. So let's look at the structure of sugar molecules. All right, so now we're going back to the web. Okay. So we're going to make a sugar called glucose. And glucose is the probably the most important sugar there is, because it makes up carbohydrates. It's the monomer of carbohydrates, and it is our and other animals, other mammals, our blood sugar. That's the sugar that's flowing through your arteries and veins, is glucose. And now sometimes you'll see glucose written in a linear way, but we're not going to do that. We're going to always put it in a ring formation. And wait a minute. Now you can draw it like this and put all the carbons, all the oxygens, all the hydrogens in it like this, or you can do the shortcut, which is this, where you just draw angles, and every angle is a carbon, and just take it for granted that there's a carbon there, and there's a hydrogen dropping down here, and hydrogen over here. Remember the molecular formula for glucose is C6H12O6. And so you just take now, and in the ring, there are 5 carbons in the ring and 1 oxygen in the ring. And 1 of the carbons is outside the ring, right? Okay. And so glucose can be also called a monosaccharide. Sugars are called saccharides. A monosaccharide is 1 sugar. Disaccharide, 2 sugars. And most of the sugar that we eat are disaccharides. Glucose is found in corn. It's really corn sugar. There's also a monosaccharide called fructose, which is fruit sugar. That's a monosaccharide. And what food chemists do when they take glucose, and believe it or not, glucose is a sugar, and sugars are sweet. But actually, glucose is not all that sweet. Fructose is much sweeter, and that's how it interacts with your taste receptors. It's much stronger sugary taste than glucose. So what they do is they take corn sugar, glucose, bring it into the lab, and change it into fructose, and then put it into sodas and sugary drinks. This is called high fructose corn syrup. I don't particularly care for it because it's made in a lab. I'm kind of particular about things that I eat. The things I want to eat are made by another organism, not made by some chemist in the lab, but that's just me. Anyway, but fructose and glucose are isomers of each other. They have the same microformula, but different structural formulas. Okay, the disaccharides that we eat, one is, table sugar, sucrose. That is actually a glucose and a fructose combined together. We also eat lactose. If you drink milk or cheese, lactose is milk sugar. And it's glucose and another sugar called galactose, which is another isomer. And then the third disaccharide you might eat is called maltose. It's grain sugar, and it's what they use to brew beer, is maltose. And we're going to actually make, well, if we were in lab, we were going to make a molecule of maltose. So in the lab paper, it wants you to draw the molecule using the shortened way. And is it a polar molecule? Does sugar dissolve in water? Oh yeah, it's a very hydrophilic molecule. And then what you do is disaccharide. You would find a partner at your table, and you two would put together two glucose molecules together in a particular way to make a maltose molecule, to make a disaccharide. The instructions are right there. And it's called a 1,4 bond. So what you do is you link the number 1 carbon with one glucose to number 4 carbon in the other person's glucose. And here's the numbering arrangement. There's 6 carbons. The carbon on the outside is the highest, it's always the highest number, so that's 6. You count backward from there. 5, 4, 3, 2, 1. So those is the numbering scheme. And it shows you there on the lab paper also. So what you're going to do is link number 1 carbon on one glucose molecule to number 4 carbon on the other person's glucose molecule, and take away water and leave an oxygen left. So let me show you what and that's called dehydration. Let me pull it up, dehydration. Dehydration reaction. Okay, here we go. 2 glucoses. There's number 4 for that glucose. There's number 1 for this glucose. And we're going to take away an oxygen here, a hydrogen, another hydrogen, leave an oxygen behind it, and that's water, that's H2O. And there's an oxygen that's linking the 1 and the 4 to make maltose. That's it. That's a dehydration reaction. And then it says, how would you make a trisaccharide? Well, you add another sugar. You either get use this 4 and link it with a 1 on another sugar on this end, or this 4 and another 1 on this end to make a trisaccharide. How do you make a polysaccharide? Add a 4th or more, and that would be a polysaccharide. And a polysaccharide is a carbohydrate. That's what it is, carbohydrate. So the monomer for carbohydrates are sugars. And where do you get these carbohydrates in your diet? What do you eat to get carbohydrates? And we want to enter a guess. And what are these carbohydrates called?
You eat carbs.
Yeah, so what kind of food would you eat to get carbs?
Bread or pasta.
Bread, pasta, potatoes. This is starch. Starch is a plant-based carbohydrate. And when you eat that carbohydrate, you have to digest it. Now that's the opposite process. That's called hydrolysis. So if you reverse this arrow and you add water, you can break apart maltose into glucose. And that's what you have to do when you eat this stuff. In order to get the nutritive benefit of carbohydrate, you have to break it into individual glucose molecules with an enzyme called amylase. And you absorb the glucose into your blood, it goes right to your liver, and your liver makes another kind of carbohydrate called glycogen, sometimes called animal starch. Right? And then it doles out the glucose between meals to make sure your blood sugar stays the same. Your blood sugar is always the same. And people are unsure about this. Like I said, what happens to your blood sugar if you skip a meal, or you don't eat it all that day, or you don't eat for 2 days or 3 days? What happens to your blood sugar?
It goes down.
You would think it'd go down, right? It doesn't. It stays the same. It's always the same. It doesn't go down. And here's why. The big guy, your brain, absolutely depend upon glucose. It has to have glucose. It can't survive on anything else. It can survive on these things called ketone bodies a little bit, but it still needs glucose. And that's why you pass out if your blood glucose goes low. That's why you pass out, because your brain's not supplied with energy. It has to have it. So if you don't eat any carbs, like in one of those diets called ketogenic diets where you don't eat any carbs, your body makes the glucose from usually amino acids, which we're going to get to make sure that your brain is supplied with glucose. So it never changes. You can be in the last stages of starvation and your blood glucose is the same. Okay. All right. So then we move on from carbohydrates to lipids. On page 5, we talk about lipids, and first kind of lipid we talk about, several types of lipid. It's called triglyceride, and this is fat. This is dietary fat, the fat that you see on a steak or on a chicken, or usually animal fat. But it's also plant fat. Also, it's plants and animals both make triglycerides. Plants make saturated triglycerides, which we're going to get to. I mean, animals make saturated triglycerides. Okay, triglyceride is 3 of something, right? Tri. So composed of a molecule called glycerol and 3 fatty acids. And so we'll look at glycerol and 3 fatty acids. So let me put that up. And is this, there are different types of lipids, and they do different things, and they are different. But the only really big thing they have in common is they're nonpolar. Even though triglyceride has a little bit of oxygen, it still does not mix. You put fat in oil and water, it doesn't mix. So glycerol is this molecule here. So what I would have students do is make a triglyceride by cannibalizing their maltose. They would talk about their maltose, and it's the whole table would think about their maltoses and make one of these molecules, one of these glycerol molecules for the whole group, and then make 3 of these. Fatty acids. There's a couple of fatty acids. They're long strings of carbon and hydrogen. They're hydrocarbons. And the question is, why is it called a fatty acid? Well, for one thing, it's acidic because it has this carboxyl group at the end here. And we know that carboxyl is an acidic group. So there's the acid part. But this part here is hydrocarbon. It's fat. The fat is just carbon and hydrogen. So this is the fatty part. That's the acid part. So that's what makes it a fatty acid. And the interesting thing is it a polar or non-polar molecule? Well, it's a little hard to say because this part, we know carboxyl group is a very polar, right? But this over here is very nonpolar. So this is called an amphipathic molecule. It's both. It has aspects of nonpolar and polar, right? And that's a pretty useful molecule to have. Soaps are amphipathic molecules. They mix with oil, but they also mix with water. Okay. Then it talks about saturation and unsaturation. And you've probably heard of these terms. If you look on food labels, you'll see how much lipid is in it, how much protein is in something, how much carbs are in something, and how much fat is in it, and how much is saturated. Right? And saturated means as much hydrogen as possible. So this stearic acid is an example of a saturated fatty acid. It's all single bonds between the carbons, and it has a maximum amount of hydrogen. This linoleic acid is an unsaturated fatty acid. Why? It has less hydrogen. And remember what you had to have if you take hydrogen away. Remember, we had a smaller molecule where you had to take away 2 hydrogens from alcohol and make a legal molecule. How did we do that? Double bonds. So unsaturated, since it doesn't have enough hydrogen to fill up all the bonds, it makes double bonds between the carbon. And if you have one double bond in the fatty acid, it's called a MUFA, monounsaturated fatty acid. If you have 2 or more, like linoleic, this would be a PUFA, a polyunsaturated fatty acid. So you got MUFAs, and you got PUFAs. So this second question down on page 5 is, what do you need to complete the molecule? A double bond. You got to have a double bond. All right. And here's the thing. What a double bond does to the molecule, it puts a bend in a crick in the molecule. Saturated fats have straight fatty acids, and they pack together real tightly. So fat, like animal fat, is mostly saturated, and it's solid at room temperature. Right? But plant fats are generally unsaturated. They have double bonds in them. There's a crick there. So they can't pack it together as closely because you got this one part sticking out here. So they're liquid at room temperature. And there's where all the plant oils come from. Olive oil, canola oil, corn oil, all these. Because plant fat is generally unsaturated, saturated fat. Animal fat is generally saturated, except for fish. Fish oil is unsaturated. And matter of fact, it's really healthy for you because it most of these fatty acids we can make, but we can't make what are called the omega fatty acids, omega-3, omega-6. We can't make them, so they're called essential nutrients. We have to get them in our diet. And that's why fish is so healthy. Also, flax seeds have this omega fatty acids. That's why these things are so good for you. Okay. The other type of fat, and I tend to go on and on about this, is what's called trans fat. Oh, wow. We know that saturated, we know since the 50s that saturated fat is the amount of saturated fat you have is a direct link to your risk for heart disease. So most dietitians recommend that you don't eat more than like 10% of your diet or less for saturated fat. You need some saturated because that's also where you get fat-soluble vitamins from. but you tend to make a small amount. I mean, you may not worry about heart disease now, but you will be, or you should be, when you get to my age. So, you worry about how much saturated fat you eat. But there's something called trans fat. Now, this unsaturated fatty acid, this is called a cis formation for this double bond. There's a different way to make a double bond called a trans formation. And that is, this crick is straightened. So it looks like this, even though it has a double bond. And this is done in the lab. It's an artificial fat. And they put it into food to increase shelf life, mostly in baked goods, like baked cookies and muffins and pastries, and has what's called trans fat. And it is absolutely terrible stuff to eat. You want to increase your risk for heart disease. Saturated fat pales in comparison to the risk you get by eating trans fat. It wasn't even supposed to be food. It was made to be to be put into candles back in the day before we had electricity. And now they put it into food because it increases shelf life. And it's something that I try to avoid. And it's just me. If I read a food label and if there's trans fat in it, I put it back on the shelf. And here's the tricky thing. It's supposed to tell you on the food label how much trans fat is in it, but they're allowed to lie. They can put 0. If there's less than half a gram of trans fat in this food, they can put 0. So what you have to do is have to look at the ingredient list. It's also called partially hydrogenated vegetable oil. That's what will be in the ingredient list if there's trans fat in there. And that's why I read the ingredient list. And if I see partially hydrogenated anything, it goes back on the shelf. Anyway. Okay. So what you got to do that 4 people at a table, what time we got? 4 people at a table would be making a triglyceride. So they make 1 glycerol and 3 fatty acids to make this. And how would you do that? Well, okay, here's the, okay, where's the monoglyceride? Well, there's a triglyceride, 3 fatty acids. There's the glycerol molecule. These are 3 fatty acids. So you're going to put these together and look at what we have. We have an OH here and OH here. What do you think what kind of a reaction do you think you're going to use to put those 2 together? Dehydration. It's the same as putting sugars together. Now, what would be the, so first thing that they'd make would be all right, let me put monoglyceride. Okay, the first thing you do, it was a good example of a monoglyceride. What's going on here? On the right, it's a monoglyceride. It's a glycerol molecule with only 1 fatty acid, monoglyceride. Then you take another fatty acid with another dehydration reaction and put another fatty acid on, diglyceride. And you've seen on food labels, mono and diglycerides. Lots of breads and things have mono and diglycerides. And then you put a third on at the left to make a triglyceride. And we would only have fatty acid chains that are 6. Most of your fatty acids in your body are like 16, 18 carbons long, but we weren't going to do that in the lab because that'd be way too much work. We would just have 6 carbon, short chain fatty acids, have 6 or 8 carbons. And so we make a triglyceride with short chain fatty acids. And well, where's the short? So Here would be an unsaturated triglyceride. If even one of the fatty acids is unsaturated, we call this an unsaturated fat, and see that cricket. So that's why it doesn't pack together as well. So this would probably be a plant triglyceride. Okay, so you now have a monoglyceride. What do you have? What would you have left over? Water. What is this reaction called? Dehydration. And how are you gonna make a triglyceride? You add 2 more fatty acids, 2 more dehydration reactions, you got a triglyceride. So that's how you answer those questions, right? Okay, last part of the lab before we answer some questions is looking at proteins. And amino acids are the monomers of proteins. And so we have to look at amino acid structure, and you make an amino acid called glycine. The first is like an amino acid structure. Okay. Amino acids have this structure. Now, ignore they are for a moment. This amine group, and this is why it's called amino acid. There's an amino group. There's a carboxyl group. Amino acid. And every single amino acid that exists have this structure right here. But what makes one amino acid different from another amino acid is what's on the 4th bond of this carbon, an R group, which could be anything, absolutely anything. So there's an immense number of amino acids out there, because that R group could be anything. But it just so happens that in proteins, which are polymers of amino acids, there are only 20 different amino acids that are used in proteins. So some people think, oh, that means there's only 20 amino acids. No, there's gazillions of amino acids. But sometime early in life on Earth, these 20 were selected by some ancient organisms to make up proteins, and we're stuck with them. All life on Earth uses the same 20 amino acids. So that makes proteins more complicated than anything else. I mean, carbohydrates are polymers, right? But what are they polymers of? Glucose, 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 glucose is one type of monomer. Amino acids, 20 different monomers, 20 different types of building blocks with 20 different R groups, right? So let's look at amino acids, amino acid list. So, and this usually comes out pretty small, but anyway, there are the 20 amino acids. They all have names that usually end in INE, like lysine, glycine, some end in acid, aspartic acid, proline. They all have different R group. Some are polar R groups, some are non-polar R groups, and so on and so forth. And we can make most of them. Some of them we can't. That's what's called the essential amino acids. Those amino acids you have to get every day because you can't make them if you want to make proteins, right? Okay, so what I would have students do is each one would make glycine, which is the simplest amino acid, just has a hydrogen as the R group. And you construct a dipeptide, that's 2 amino acids, by a dehydration reaction again. So, and you make something called a peptide bond. So let me show you that. Okay. You're gonna put these 2 amino acids together. So the blue is an OH attached here on a carboxyl group, an H on an amino group Take away, that's water, and here is a peptide bond between a nitrogen and a double bonded carbon oxygen, peptide bond. Then how would you make a tripeptide? Add another one. Add another amino acid with a nitrogen group over here to this carboxyl, or carboxyl with an amino acid on this side to make a tripeptide. Polypeptide, add a fourth and more. And a long chain of amino acids makes up a protein or polypeptide. And the order of the amino acids is called the primary sequence or primary structure of the protein. And every protein has a different primary structure. And so this means they fold in all different ways depending upon their primary structure and into a three-dimensional shape. And that three-dimensional shape is really important because most proteins work by binding to other molecules. They have to have a complementary shape. And their shape is determined by what amino acids are put in what row. And how do your cells know how to make an amino acid? How do they know how to put glycine here and proline here, aspartic acid here? This is the information in a gene. Genes are recipes, most genes are recipes for proteins. What amino acid do you put in what order to make a specific protein? Right? And I think that's it for that. Let me go back to the molecules lab. So you make a dipeptide, and a tripeptide is 3, and then polypeptide is 4 or more. Okay. So we're going to go on and answer these questions. Anybody have any questions or any problems with the stuff so far? All right.
I have a question.
Sure.
I was wondering for the questions where it's asking us to draw a molecule, do you want us to like find a picture that we can add to the document, or should we like actually draw them and then?
Yeah, you could actually take a pencil or pen and actually draw them using, you know, the letters for the atoms and, you know, lines between them for the bonds. Or if you're going to draw a sugar, you can just draw, you know, the angles for the carbons, and you do the shortened form if you want.
Okay, thank you.
Okay. All right, let's answer his questions, and then we're out of here. We still got an hour to go. What are the differing on page 6, the last page? What are the different physical and health attributes of saturated compared to unsaturated fatty acids? All right. What's the physical difference between a saturated fat and an unsaturated fat? So saturated has as much hydrogen as possible. Unsaturated doesn't. So what do you need? So what does a unsaturated fat have that an unsaturated fat have that a saturated fat does not have? Remember what you need when you take away hydrogens between the carbons. What's there?
Double bonds.
Double bonds. So that's the physical difference between saturated and unsaturated fatty acids. And also, you know, in cis formation, unsaturated has a bend in the molecule. Transformation, it doesn't, but natural fats, actually there's a few natural trans fats, but they're very rare. Mostly, yeah, so the most natural unsaturated fats have a bend in the molecule. Okay. And then the health attributes, as I mentioned before, they've known since the 50s in what's called the Framingham Heart Study that the more saturated fat you eat, the higher your risk. Doesn't mean you're going to get a heart attack or a heart disease, but it just increases your risk for it. That's all. So you like your double bacon cheeseburgers now, but after a while, you might want to ease off on those. Anyway, but then you go on to carbohydrates, and actually, I should have mentioned this before in carbohydrates. I said carbohydrates are long strings, 4 or more, but that's really, in food labels, that's called complex carbohydrates. So like starch is a complex carbohydrate, but they call even disaccharides carbohydrates, but they call them simple carbohydrates, all right? Even monosaccharides are called carbos, but they're simple carbs versus complex carbs. And the thing is that the simple, a lot of doctors and nutritionists now think what's even worse than lots of saturated fat is lots of processed carbs. Now, processed carbs are carbs made by taking all the nutrients away from a perfectly good food and having it just pure carbohydrate. There's a saying made by one guy where it says, the whiter the bread, the quicker you'll be dead. So the processed carbs are the white foods like white bread, white rice, white pasta. These are processed carbs, and a lot of people think that they're more dangerous for you than eating a lot of fat acids. You want to eat processed carbs that are full carbs, that have other nutrients in there rather than just pure carbohydrate. But what is one biological function of carbohydrates? What do we say carbs do for us?
They provide energy through glucose.
Right, and the time frame is like, when you think of sugar, you think of quick energy, right? Well, carbohydrates, we have a carbohydrate storage molecule called glycogen, and it's for maintaining blood glucose between meals. It's for short term, between meal, maintain a blood glucose. So that's one thing. So a short term energy storage is one form of one function of carbohydrates. But in another function of carbohydrates that is found in plants is cell walls. They use cell walls, a carbohydrate called cellulose, to make their cell walls. All right, and this carbohydrate is actually very beneficial to us if we eat it, even though it's non-nutritive. And this is called insoluble fiber. It's a cell wall. It's a long string of glucose molecules, very similar to starch, but it's not digestible. We can't digest it because it has 1,4 bonds, but it's shaped slightly different than the 1,4 bonds in starch. It's called an alpha bond, and the 1,4 bonds in starch are called beta bonds. We have enzymes that can break the beta bonds and absorb that sugar. We don't have the enzyme to break the alpha bonds. Now you can, you do digest it to a certain degree, like a stick of celery. It's like 99% cellulose, and you start chewing it, and you can chew it all day long, but you're not going to break it into individual monosaccharides. And in order to get the nutrient benefit from it, you'd have to do that. So you chew it, it goes into your stomach, into your intestine, and it just stays there. We can't digest it. So it eventually leaves your body. It looks real different when it leaves, but it's the same molecule. It's cellulose. So why do people, doctors and dietitians say, hey, it's good to eat your fiber, eat this seemingly useless stuff? Oh, somebody in the waiting room, somebody got kicked out. Why should you, why would it be healthy to eat this seemingly useless stuff? Well, it's not completely useless. It moves things along. You really have to move things along in your GI tract, right? Regularity, you want to be regular. It's good to defecate at least once a day. If you eat things that are completely digestible, you probably won't be doing that. And so it's really good to move things along. So that's why people, say you should eat your fiber. It also fills you up. Now, one of the problems is in the US that we tend sometimes to eat too many calories. I mean, calories are one time tough thing to find when, thousands of years ago when we were hunter-gatherers. Tough to find, but now it's easy. And we tend to eat too many calories. But if you eat a lot of fiber in your food, it fills you up and you eat less calories. And that's usually a good thing, not to eat too many calories. So that's some function of carbohydrates. Even the indigestible carbohydrate has a important biological function. Okay. Next, biological function, triglycerides. What does fat do for you? Anybody? It's similar to carbohydrates.
Doesn't fat store energy for later?
Yeah, that is energy storage as well. Your adipose tissue is filled as fat cells filled with triglycerides. And right now you're living on really both carbs and fat. But the more you go out eating, the more you'll switch to fat. And so, like I said, if you stop eating for a couple of days, you're going to starve? No. You're going to be breaking down your fat stores. And everybody's got fat stores. Even the thinnest person's got enough fat to last a month or a couple of months without eating. So it's long-term energy storage is one biological function of triglycerides. The other is insulation. It's really good insulation. Two types of insulation. One, thermal insulation. We have a layer of subcutaneous fat over our whole body underneath our skin, and it helps to insulate you against the cold. It's good insulation, thermal insulation. But it's also electrical insulation. Now we have plastic and rubber around wires in your house. We have wires in our body called axons, called nerves, and they are surrounded, most of them anyway, are surrounded by a fatty covering called myelin, and it's fat. And so it's like the plastic or the rubber around wires, it's electrical insulation. So that's important role for fat as well. Finally, what is one biological function of proteins? Well, proteins are the most complicated and have the most roles of anything else. They do all kinds of things. Sometimes it's as simple as structural protein, to hold things together. Enzymes. We're going to do a lab on enzymes. The first lab back on campus could be enzyme lab. And those are essential for life. You have cell ID proteins that identify you as who you are. There's membrane transport. And there's transport proteins and there's membrane transport proteins. There's receptors there's signaling molecules like hormones, and there's receptors that bind to the hormones, and they're proteins also. So there's all kinds of roles. And one role actually is energy yielding. You can talk about carbohydrates and triglycerides as what's called energy yielding macromolecules. We make ATP from those molecules, and we get energy from them. Proteins, you can get energy from also. You can break proteins down to get ATP, but we generally don't. that we use the carbohydrates and the fat first. And then if you go long enough without eating, then you'll start breaking down protein. We start breaking down your own muscle tissue to make glucose to keep your brain alive. But now you're in like the last stages of starvation. But you can also, if you eat more protein than you need, and we actually you don't need that much protein. Most of us eat more protein than we really need to make the enzymes and receptors that we need to make. And so if we eat more protein than we need, and actually if you eat more carbs than you need, because you have this glycogen gas tank in your body, and when it gets filled up, you can't put any more in. So those are converted to fat. Eat more protein than you need, eat more carbs than you need, you make fat out of it, you store it for long-term storage in fat tissue, and you put on some weight. Okay, I think that's it. And it's not even noon yet. So let me stop screen sharing and answer any questions you got about, well, anything that we pretty much do at the lab.
I have a question.
Sure.
So I took my onboarding test for the exam, and they said there's two onboarding tests on the website.
Yeah, I don't know why there's two of them on there. That must have been my error or something. You just take one of them.
Okay.
Take one. As soon as you get cleared on that, then you're cleared to take the exam.
Will the exam open today, or will it open later this weekend?
I think it'll be open a little later, maybe tomorrow, maybe this weekend. And I'll put up an announcement when I've already got it written up. It's ready to go. So it shouldn't take me too long. And I'll put an announcement in about it.
Okay.
About how to take it, when to take it, when it closes, and everything associated with it. Okay.
In that exam, it's on the first 3 chapters, right?
Correct. Yes. Okay, I'm going to stop recording.